Hello, I'm Dr. Ben Whittaker, a postdoctoral fellow in the Department of Integrative Biology at the University of Guelph. Today I'm presenting a talk for the Ontario Ecology, Ethology and Evolution Colloquium in collaboration with co-authors Sumika, Maeda and Professor Elizabeth Boulding. Our presentation is titled, What Did the Cleaner Fish Say to the Salmon? Communication by a facultative cleaner wrasse facilitates interaction with infected Atlantic salmon. Before I begin, we would like to acknowledge the Natural Science and Engineering Research Council of Canada for funding this project. We thank Cook Aquaculture, Kelly Cove Salmon Limited, the Huntsman Marine Science Centre and ProNova Marine Products for contributing the animals used within this study. We would also like to thank the staff of Hagen Aqualab and the volunteers, students and staff of the Boulding Research Group for their help throughout the study. Professor Edward Wilson, widely regarded as the founder of sociobiology, defines animal communication as occurring when a cue given by one organism, known as the sender, is perceived by and alters the probable behaviour of another organism, known as the receiver, in a manner that is adaptive for one or both of the participants. Fish have developed numerous strategies for communicating with each other, including producing grunts, purrs and clicking sounds by using their swim bladder. Some groups, like the knifefish, have evolved specialist organs that produce electric pulses for communication. Herring even emit gas to communicate, allowing flatulent messages to pass through the shoal undetected by their predators. Some fish have evolved highly sophisticated methods of communication, which allow them to occupy unique niches within aquatic ecosystems. The bluestreak cleaner wrasse is a small species of tropical marine fish found in coral reefs across the Indo-Pacific. This cleaner fish feeds on the ectoparasites of other species of fish, known as its clients. This is an example of mutualism, as the fitness of both parties is mutually benefited through interaction. The client has its parasites removed, and the cleaner gets a meal. Bluestreak cleaner wrasse are classified as a dedicated cleaner fish, because they satisfy the vast majority of their diet through cleaning clients. This means they need to interact with many species of fish, representing diverse species, in order to find enough parasites to meet their daily energy needs. When it comes to fish communication, the bluestreak cleaner wrasse are top social influencers and use multiple forms of communication to facilitate safe and prolonged cleaning interaction. However, this mutualism is not as peaceful as it first appears. The bluestreak cleaner wrasse has weaponized its communication strategies and occasionally uses its influence to deceive other fish, switching to parasitism by taking a bite out of its client's flesh. This manipulation demonstrates the Machiavellian intelligence of the species, and the interaction between bluestreak cleaner wrasse and clients has become a model system for understanding the evolutionary game theory behind cooperation and cheating strategies in nature. Let's discuss some of the interactions that occur between bluestreak cleaner wrasse and their clients. First, the cleaner and client may advertise their willingness to engage in cleaning interaction. Solicitation occurs when either of the fish adopts a conspicuous pose, such as flaring the fins and opercula, or swimming in a headstand or tailstand posture. This brings the cleaner and client into closer proximity for further cleaning interaction. The cleaner may then show tactile stimulation, which is an oscillating dance-like movement with close body contact to the client. This massage-like stimulation lowers client aggression, prolongs the duration of cleaning, and acts as a positive reinforcer for future encounters. Posing and body contact cues are forms of communication, as the expression of these cues alters probable behaviour in their partner, both in terms of closer proximity and sustained interaction. Cooperative interaction takes place when the cleaner searches the client's skin for parasites, often with the client remaining motionless to allow close visual inspection. If the cleaner finds a parasite, it is removed with a gentle pecking motion that does not injure the client. Signs of antagonistic interaction occur immediately after the cleaner cheats by biting the client. This involves the client suddenly and rapidly jolting away from the cleaner to end interaction. Some clients respond to cheating by chasing the deceitful cleaner, which acts as a form of punishment. Interactions between the dedicated class of cleaner fish and clients are well documented in scientific literature. However, the vast majority of species which engage in cleaning behaviour do not belong to this category, and are instead classified as facultative cleaners. These species show great variation in the extent of cleaning behaviour and do not rely on eating parasites to the same extent as dedicated cleaners, often foraging for alternative prey items. There are numerous species of facultative cleaner wrasse. Some of these are only distantly related to the bluestreak cleaner wrasse and are found in cold, temperate regions, such as the European ballan wrasse pictured on the top left. Numerous species of goby, shrimp, and the juvenile stages of several angelfishes also engage in facultative cleaning. Facultative cleaner fish come in surprising forms, such as the miniature stickleback and the almost spherical lumpfish pictured on the bottom right. Information on how these facultative cleaners interact with clients is scarce, with only a handful of studies straying from the tropical dedicated species. This limits our understanding of cleaning interaction, communication strategies, and the trade-offs between mutualism and parasitism. It also hinders the use of facultative cleaner fish in commercial salmon aquaculture. Lice infestation is a significant problem on salmon farms, as the high stocking densities associated with intensive fish production create the ideal environment for lice populations to thrive. Lice infestation causes lesions on the salmon's skin, increases mortality, and spreads disease. Additionally, the lice can disperse from the farm and infect wild fish in the surrounding ecosystem. These lice threaten sustainability of salmon farming by creating economic loss, ecological damage, and serious concerns for fish welfare. European salmon farms have deployed facultative cleaner wrasse into farms since the 1980s. The industry now uses 5 species of wrasse, and each has been shown to help reduce the number of lice on salmon inside farms. However, the exact nature of how these wrasse remove lice and whether this involves communication is unknown. The cunner wrasse is the only cleaner wrasse that has been trialled in Canadian salmon farms. We know even less about this species than we do about the European wrasses, and understanding how this fish interacts with salmon may provide valuable insight for developing sustainable parasite control in Canadian salmon farms. We aimed to assess how cunner wrasse interact with Atlantic salmon and whether the two species exhibited communicative behaviour. We hypothesised that a behavioural cue, such as posing or body contact, might constitute interspecific communication if its perception alters the probable behaviour of the receiving fish in an adaptive fashion, for example by facilitating cleaning mutualism. To test this hypothesis, we made 4 predictions. First, that behavioural cues should be less common when the salmon are not infected with lice, as neither the cleaner nor the client benefit from interaction without parasites. Second, that increased expression of behavioural cues should reduce the distance between cunner and salmon, as this allows for further cleaning activity. Third, that increased expression of behavioural cues should increase mutualistic cooperation, such as inspection for parasites and removal by gentle pecks. Finally, we predicted that increased expression of behavioural cues should decrease antagonistic behaviours, such as jolting and chasing, as these would end cleaning interaction. To test our predictions, we recorded the interaction between 1 cunner wrasse and 3 Atlantic salmon for 30 minutes. Each of our 43 cunner wrasse undertook the trial twice, once with salmon that were infected with Calmus stage sea lice and once with non-infected salmon. We then coded videos in the Boris software package using an ethogram that contained all our predefined focal behaviours. Although we had planned to record chase behaviour, this was not observed in any of the trials and so was not included in analysis. We analysed video stills taken every 5 minutes in ImageJ software and used known reference points to calculate the average distance of cunner from each of the salmon. We analysed data in R by using a series of linear mixed effect models and generalised linear mixed effect models fit with Poisson distribution to test each prediction, depending on the nature of the response variable. Graphs 1a and 1b show the changes in frequency of behavioural cues for each cunner in the presence and absence of parasites. On each graph, a positive value indicates greater frequency in the presence of parasites. A negative value indicates greater frequency in the absence of parasites, and a value of 0 indicates no difference between the trials. The frequency of body contacts are shown on the left-hand graph and posing on the right. The individual cunner are ordered by increasing weight along the x-axis. We found a significant association between the presence of lice and the frequency of pose behaviour. We also found that larger cunner showed significantly higher frequencies of contact behaviour when lice were present. Graphs 2a and 2b show the average distance between infected salmon and cunner at 5-minute intervals throughout the 30-minute trial. The frequency of behavioural cues expressed during each of the 5-minute intervals is shown on the x-axis, with posing shown on the left-hand graph and body contact shown on the right. We found greater expression of both behavioural cues was significantly associated with closer proximity of salmon and cunner. Graphs 3a and 3b show the total time duration each cunner inspected infected salmon and the frequency of each behavioural cue, with pose behaviour shown on the left-hand graph and body contact on the right. Analysis showed a significant increase in the inspection times with increased expression of each behavioural cue. We did not establish any significant effect of behavioural cues on antagonistic jolting behaviour. However, there was a high degree of behavioural variation in the interaction between individual cunner and their clients, and we found that total interaction frequency was significantly higher for larger cunner. These results support our first three predictions. We found expression of posing and contact behaviour was lower when cunner were with parasite-free salmon, that these behavioural cues reduced the average distance between cunner and infected salmon, and that cue expression increased inspection duration. However, we did not establish any association between the cues and antagonistic behaviour, perhaps because jolting and chasing were rarely observed. We also found a high degree of individual variation, suggesting cleaning behaviour is not a species-wide behaviour that is expressed by every cunner. However, larger cunner were more likely to show cleaning interaction. Our study is among the first to investigate facultative cleaner interactions with clients, and more research is needed before we are able to draw firm conclusions on the nature of this behavioural interaction. As facultative cleaners forage on alternative prey items, it might be expected their ability to remove parasites is limited. However, our results imply that facultative cleaner fish show scope for sophisticated client interaction. We provide evidence for possible interspecific communication between a facultative cleaner wrasse and its clients, implying parallels with dedicated cleaner fish that may warrant further review of how cleaner species are classified. Our study found low rates of antagonistic behaviour, which would be beneficial on salmon farms. However, we found a high degree of behavioural differences that suggest facultative cleaning is not displayed by all individuals within a species. Our study suggests that salmon farmers should use larger cunner to help control parasitic lice, as these are most likely to clean salmon. Ongoing work in the Boulding lab aims to develop husbandry protocols, improve welfare, and accelerate growth rates of cunner wrasse, providing a reliable supply of sustainably sourced cleaner wrasse for Canadian salmon farms. The high degree of individual variation in cleaning activity may present an obstacle to industry. The Boulding lab also aims to better understand the sources of this variation and develop tools such as behavioural screening protocols and assessing genetic heritability of traits to provide new methods for increasing cunner wrasse efficiency in farms. Although this pioneer study shows promise, more work is needed to evaluate how these cleaning interactions occur in a farm environment, especially whether posing and contact behaviour is correlated with parasite removal rates. We are only just beginning to translate what the cleaner fish said to the salmon. For more information, please refer to our publication in Applied Animal Behaviour Science. You can read more about the study in our article published in The Conversation. Please feel free to reach out by email or social media with any questions.